Good morning, students. I am Anita Amani, Associate Professor at GCW Lakanmajra, Rohtak. Today's our topic of discussion is electron transport chain. The electron transport chain or the electron transport system is the mechanism that a cell uses to convert the energy from NADH and FADH2 into ATP. Electrons flow along the energy gradient via carriers in one direction from high reduction potential to lower reduction potential. The ultimate acceptor is oxygen, molecular oxygen in aerobic respiration or sulfate in anaerobic respiration. In electron transport chain, electrons are passed from one molecule to another and energy released in this electron transfer is used to form an electrochemical gradient. In chemoosmosis, the energy stored in the gradient is used to make ATP. ATP is used by the cell as the energy for the metabolic processes for cellular functions. In the process, protons are pumped from the mitochondrial matrix to the intermembrane space and oxygen is reduced to form water. Electrons transport chain is also called cytochrome oxidase system or as the respiratory chain. ETC produces 34 ATP molecules compared to 2 ATP for glycolysis and 2 ATP for Krebs cycle. The main biochemical reactant of ETC are the electron donor succinate and NADH. ETC or electron transport chain is present in the inner mitochondrial membrane. Mitochondria is also called powerhouse of the cell because all the energy is produced in the mitochondria, and the enzymes for the ETC are located in the inner membrane. You can see the components of ETC, that is electron transport chain, that is NAD, nicotinamide, adenine dinucleotide, FMN and FAD. FMN means flavin mononucleotide and FAD is flavin adenine dinucleotide. Then there is ubiquinone or coenzyme Q, the iron sulfur proteins, cytochrome B, C1, C, and A3, and the other complexes. All these are the components of ETC. This is diagrammatic representation of various components of ETC, that is electron transport chain. Complex 2, it is also called succinate dehydrogenase, contains iron and succinate which oxidizes FAD to form FADH2. Complex 1, also called NADH dehydrogenase, it contains FMN which accepts 2 electrons and hydrogen ions from 2 NADH to become the reduced form of FMNH2. Also contain iron atoms which assist in the transfer of electrons and hydrogen ions to coenzyme Q. Coenzyme Q is well defined as crucial component of oxidative phosphorylation process in mitochondria, which converts the energy in carbohydrates and fatty acids into ATP. Coenzyme Q or ubiquinone accept electrons from FMNH2 from complex 1 and FADH2 complex 2 and transfer the electrons to complex 3. Next component of the electron transport chain is complex 3, also called cytochrome reductase. It contains heme group in which the Fe3 positive ion accepts the electron from coenzyme Q to become Fe2 positive ion. Then it transfers the electrons to the cytochrome C. Cytochrome C is essential component of the electron transport chain where it carries one electron. It is capable of undergoing oxidation and reduction as its iron atom converts between ferrous and ferric forms. It then transfers electrons to complex 4. Complex 4, it is also called cytochrome A, cytochrome A3 complex. It contains the heme group in which Fe3 positive ion accepts the electron from cytochrome C to become Fe2 positive ion. Complex 4 transfers electrons to oxygen, which combines with hydrogen to form water. This next component of ETC chain is called complex 5, or mainly we call it ATP synthase. It contains a proton channel that allows for the protons to cross into the matrix using the proton gradient energy to catalyze the synthesis of ATP from ADP and phosphate. Good morning, all of you. This is Anita Mani, Associate Professor at Government College for Women, Lakhan Majra, Rohtak. Today we are going to discuss the TCA cycle, that is tricarboxylic acid cycle. We also call it citric acid cycle or Krebs cycle. Citric acid cycle is actually a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl coenzyme A derived from carbohydrates, fatty acids, and proteins. It is second stage of cellular respiration. This process is carried out in matrix of intracellular structure called mitochondria, except in bacteria. Krebs cycle or citric acid cycle is one of the most important reaction sequences in biochemistry. Citric acid cycle is closed loop. The last step of pathway regenerate the molecule used in first step. Krebs cycle starts with the product of glycolysis, which are 2,3 carbon molecules called pyruvate. This molecule is acidic, which is why the Krebs cycle is also called TCA, that is tricarboxylic acid cycle. Throughout a number of reactions, these molecules are further broken down into carbon dioxide, water, and energy. Energy from the molecule is moved to other molecules called electron carriers. These molecules carry the stored energy to the electron transport chain, which in turn creates ATP. TCA cycle consists of 8 steps catalyzed by 8 different enzymes. The cycle is initiated when acetyl coenzyme A reacts with oxaloacetate to form citrate and release coenzyme A(SH). Now let's study the reactions of TCA cycle. In step 1, there is oxidative decarboxylation of pyruvate to acetyl coenzyme A by PDH complex. This step is connecting link between glycolysis and TCA cycle. Oxaloacetate combines with acetyl coenzyme A to form citrate catalyzed by enzyme citrate synthase. In step 2, citrate is isomerized to isocitrate by the enzyme aconitase. This is achieved in a two-stage reaction of dehydration followed by hydration through the formation of an intermediate cis-aconitate. In step 3, isocitrate loses a molecule of CO2 and then undergoes oxidation to form alpha-ketoglutarate by enzyme isocitrate dehydrogenase. Along with CO2, a molecule of NADH is also formed. In step 4, alpha-ketoglutarate loses a molecule of CO2 and is oxidized to form succinyl coenzyme A by alpha-ketoglutarate dehydrogenase complex. At this stage, a second NADH molecule is also produced. In step 5, succinyl coenzyme A is enzymatically converted to succinate. It is accompanied by conversion of GDP to GTP and GTP to ATP by enzyme nucleoside diphosphate kinase. In step 6, succinate is oxidized to fumarate by enzyme succinate dehydrogenase. This results into formation of FADH2. As you can see that in step 7, it involves conversion of fumarate to malate with the addition of water in presence of enzyme fumarase. In the last step, malate is oxidized to oxaloacetate by malate dehydrogenase and closes the cycle. A third NADH is synthesized at this stage. Oxaloacetate regenerated here combined with another molecule of acetyl coenzyme A and continue the cycle. This is complete TCA cycle. All the steps are being represented very clearly. It is a very simple cycle to learn. Thank you so much.